So it's time to start the joint webinar for today. Wish to welcome everybody to this joint webinar between the International Eosinophil Society and the European Mast Cell and Basophil Research Network. So today's theme is very exciting. It's single cell dissection of human mast cells, basophils and eosinophils, so the cells that we all love to work with. My name is Gunnar Nilsson. I'm the president of the European Mast Cell and Basophil Research Network. So we is a network gathering scientists that are interested in the biology of human or mouse or any other species mast cells and basophils. And we have biannual meetings, webinars, and other activities to discuss the research around these two interesting cells. So today we're very happy to have this webinar together with the Eosinophil Society, and I hand over the word to Professor Mark Rothenberg.
Good day, everyone. It's a true pleasure to be here. I'm the president of the International Eosinophil Society. Our society is an organization of scientists and clinicians interested in the eosinophil. The society sponsors biennial meetings, monthly webinars like this one, and other educational programming for its more than 300 members. We review new information about the eosinophil and its role in health and disease. I encourage you to visit our website. We're very pleased this year to now be launching a dual membership between the EMBR and NIS. So what this means is you pay for one membership and you get two. And we're looking forward on both sides to expand our membership, our interests, and our interactions. We encourage you to join and take advantage of this opportunity. Please visit our website, and the QR code is provided for information. This was just released, so grab it while it lasts and benefit from it. And we certainly welcome ideas about this and other ways to interact better. Next slide, please. Wanted to let everyone know that we are hosting our biennial meeting this summer. It's the 13th Biennial Congress of the International Eosinophil Society, which will be held in July on the 7th to the 11th in Montpellier, France. Got an outstanding program, a lot of new innovative ways of meeting this time, including a strong focus on early stage investigators and trainees, special seminars and webinars and meetings designed just for them. A lot of educational programs. The abstracts are now being accepted for the meeting, and also offering a number of scholarships for people to attend the meeting, depending upon their circumstances. We're also considering that, in terms of their economic needs, but certainly a number of travel grants will be provided for people submitting abstracts. So this is an exciting meeting, and I hope to see you there, especially taking advantage now of the joint membership, which gives you discounted registration rates for the meeting. And please visit our website, which goes over the exciting program that's now been developed. Next slide. I want to thank the Corporate Advisory Council that's supporting the International Eosinophil Society. These are listed on this slide. This is very critical for us, and we thank these organizations, particularly these companies, for their strong support. Now I'm going to pass this on to Silvia, who's going to be one of our outstanding moderators to go over the logistics.
Thank you, Mark. So I'm Silvia Bulfone-Paus. With Roma, Sehmi, I'm going to moderate the session. And just before we start, a couple of instructions. So please use the question and answer button to ask questions, and you can do that anytime during each presentations. And I will then collect the questions and read them to the speakers. All attendees are muted and the chat feature is disabled. The webinar will be recorded and will be posted on the IES website. And To view closed captioning or live transcription during the webinar, you can click the Show Captions button. And with that, I think we can start, and I will pass to Roma.
Thank you, Silvia and Mark. In the vein of the fostering a crosstalk between the worlds of mast cells, basophils, and eosinophils, our talk title today is centered around single cell dissection of human mast cells, basophils, and eosinophils. And our speaker lineup includes Dr. Dan Dwyer, Dr. Joakim Dahlin, and Dr. Netali Ben-Baruch Morgenstern. And our first speaker today is Dr. Dan Dwyer, and he'll be talking to us about mast cells. Dr. Dwyer received his BA in biology from Amherst College and his PhD in immunology from Harvard University, where he studied roles for B cells and granulocytes in initiating allergic inflammation. He continued his studies on type 2 inflammation during his postdoctoral research with Dr. Nora Barrett at Brigham and Women's Hospital, defining the transcriptional heterogeneity of murine connective tissue mast cells and using single-cell RNA sequencing to map the cellular contributions to human nasal polyposis and define mast cell heterogeneity within this disease. I hand it over to you, Dr. Dwyer.
Great. So thank you so much for this opportunity to speak to you today about some work that our laboratory is doing in trying to understand mast cell heterogeneity and use single-cell RNA sequencing as a tool to really dig into understanding the phenotypes of mast cells that exist in human. So in 1986, Iranian Schwartz published this landmark study showing that human mast cells in different tissue microenvironments have really discrete protease expression profiles. So they did immunostaining with tryptase and chymase, finding that when you look in a tissue like the skin, pretty much every mast cell co-stains for both of these proteases. But when you look in mucosal tissues, there's a very different pattern. In the submucosa, there's co-staining for tryptase and chymase, but the mast cells that were present in the epithelial region of the tissue really stain for only tryptase. And this led to the kind of nomenclature that to this day we really use to describe mast cells in humans, where we talk about MCTCs as mast cells co-expressing tryptase and chymase, and these are found in the skin, submucosa, as well as really all peripheral connective tissues, and the MCT, which has tryptase alone, and that's the mast cell population most associated with the epithelium. Now, about 20 years later, the Schwartz lab did another study where they identified CD88, the C5a receptor, as a surface marker for MCTCs. And in one of their figures from this paper, they made a really interesting observation that there is certainly at least one more level to heterogeneity in human mast cells. So they sorted out MCTCs from the skin and the lung, as well as MCTs from the lung. And what they could see is that MCTCs from the lung were really much more able to granulate in response to Fc-epsilon-1. Both MCTC populations reacted to C5a and compound 4880. But when they looked at the ability of these different populations to generate cysteinoleucotrienes, right, pro-inflammatory eicosanoids derived from arachidonic acid, there was a really different pattern between the two compartments. So within the lung, MCTs were able to make quite a bit of cysteinoleucotrienes, MCTCs a lesser degree. But there was really no production of this mediator from MCTCs in the skin in response to any of the signals they looked at. So here we have at least one more layer of complexity, saying that maybe this TTC paradigm, it doesn't really fully encompass the phenotypes that we see. Now, the MCTC paradigm becomes even more complicated when we consider human disease. And this is where really a lot of this paradigm breaks down. So we start to see MCTCs in places where they didn't used to be. They'll be present in the airway smooth muscle, in the lung, in asthma. But you can also find them in the bronchial epithelium in asthma, as well as in the glandular epithelium in nasal polyps, right? Places that you would normally associate with an MCT phenotype. MCTs also kind of change their protease profile when we think about allergic inflammatory diseases. The protease CPA3 is really absent at the protein level from MCTs. in homeostatic conditions, but it is present within these cells in the epithelium in asthma, in the epithelium of nasal polyps, as well as in eosinophilic esophagitis. So we still use the TC nomenclature to describe these populations, and it's but I think it is important to recognize that this underscores the fact that we really don't have a great understanding of human mast cell heterogeneity, and it really goes beyond just kind of these two proteases. So a useful source for studying human mast cells within the context of disease that we have access to at Brigham and Women's Hospital are human nasal polyps. On the left is a picture I took of some polyp tissue that we received in the lab. So you can see that we get quite a bit of tissue. And on the right is a flow plot that from mast cells in a digested nasal polyp. And as I hope you can appreciate, there's a huge number of mast cells present within a nasal polyp. Coming from a mouse background, I was extremely excited the first time I digested a polyp and I found 250,000 mast cells in there. But there's also immediately apparent that there is this level of heterogeneity present. There's a big spread in Fc-epsilon-R1 and CD117 across the mast cells in the nasal polyp. And we were able to see that there was also a huge difference in mast cell concentration across human diseases. So if we look at control sinus tissue, we see that there's very few mast cells. Those increase in nasal polyps, and in aspirin-intolerant nasal polyps, which is this really severe endotype of the disease, we see a much larger population of mast cells where they can make up, say, 3 to 5% of the total cells in the tissue. So a few years back, we did a study where we flow sorted mast cells from 6 donors for single-cell RNA sequencing using the seqWell platform, which was developed by our collaborators in Alex Shalick's lab at MIT. I did see that there was an advanced question about platforms. For mast cells, platforms end up not really being a major consideration. We're fortunate in that mast cells are really well captured on every platform we've looked at. The generation of the platform ends up being the major distinction rather than the platform that is used. So when we looked at mast cells in the nasal polyp, what we saw is that rather than being kind of discrete populations, a really well-separated MCT and MCTC, instead, pretty much every marker that we looked at was expressed along a gradient. So on one end, our blue cluster here, the MC1, we saw transcripts associated with MCTCs, chymase and cathepsin G. On the other end, we found a bunch of transcripts that we didn't really think about in the context of mast cells, such as IL-17RB, which is the receptor for IL-25, and IL-17B, or GPR183, which is an oxysterol receptor. We ended up being able to link these to the MCT that were expanded within the nasal polyp. And when we set the gene scores associated with these two ends of the spectrum against each other, we could see that they really did seem to be mutually exclusive. A mast cell either expressed the blue MCTC program or the red MCT program, or we had our population in the middle that didn't really express either, that we hypothesized were a population that was kind of choosing where it wanted to differentiate into. And this mutual expression really suggests a process along the lines of polarization, where a mast cell is choosing its fate and then turning on one of two mutually exclusive gene programs within this tissue. So the minor population that we identified sitting in the corner ended up expressing a transcriptional program associated with cell cycle genes, suggesting that proliferation was a major contributor to mast cell expansion within the polyps. We were able to confirm that proliferation via flow and see that proliferation was increased as we went up in disease severity, sort of paralleling the difference we saw in mast cells. And we also saw there was this correlation between peripheral blood eosinophilia and mast cell proliferation, which may hint at some of the signals driving this proliferation. But this is still not a process that we understand particularly well. We saw a similar pool within the mast cells in eosinophilic esophagitis in a study that we did in collaboration with the Rothenberg Laboratory at Cincinnati Children's, and that was with Netali, who you'll be hearing from later on today. And when we looked at the esophagus, we could see that there was also this population of proliferative mast cells. They seemed to be enriched in active EOE relative to remission and completely absent from control. And Netali did some very nice immunofluorescence where she was able to quantify that proliferation and found that there is this really significant enrichment in mast cell proliferation within active EOE relative to remission, and also within remission relative to control. So we do think that proliferation is an important contributor to mast cell expansion. So our collaborators in the Shalek lab were also working on a separate study on the skin using the same platform. So we were able to kind of take advantage of this to compare MCTCs across compartments at the transcript level. So we noticed that there was a transcriptional module that was associated with MCTCs in both the skin and the nasal polyp, and this included things like chymase and cathepsin G, but also adhesion receptors like ICAM1, the prostaglandin the E2 receptor number 4. But there were also clearly discrete modules associated with MCTC in the skin that were not expressed in the nasal polyp, and vice versa, ones in the polyp that were not in the skin. Really notable amongst those ones enriched in the skin was MRGPRX2. Right, this is the receptor for the classic mast cell activating compound 4880, but also neuron-derived substance P. And this is really robustly expressed by mast cells in the skin. We can see that by flow. It ends up being a sort of a better marker for them almost than FcεR1. But in the nasal polyp, we see that there's really no expression whatsoever. We saw something similar with the C5a receptor, CD88, that was robustly expressed at the transcript level in the skin, but not so much in the nasal polyp. In the polyp, we saw a lot of transcripts associated with activation, chemokines, and growth factors, but we also saw things like the C3a receptor being enriched there. So there is, at least within the MCTCs, clearly this additional level of complexity that exists. So a recent study that was published in the JXMed, I think, further underscores this concept that mast cell heterogeneity goes far beyond the absence or presence of chymase. And in this study, the Tauber and colleagues used the Tabula sapiens dataset, which is sort of this compendium of digested cells from all sorts of different tissues in human. And they were able to computationally separate the mast cell cluster. And when they reclustered that, they noticed that there was kind of this range of different mast cell populations that existed. And now, interestingly, while chymase was associated with one of these clusters, the MC5, this wasn't the population that was enriched in the skin. Rather, these MC5s were mostly found in the lymph node and the vasculature. The skin was enriched for other populations that didn't really have chymase as a marker gene, even though the skin is the site of the prototypical MCTC. So I think, again, that this tells us there's just so much more to mast cell heterogeneity than we really understand. And I think it's also a study like this is really just kind of the tip of the metaphorical iceberg. As platforms and computational tools continue to evolve and new data sets are generated, I think that I will expect that we're going to see like an ever increasing number of mast cell populations in humans, especially associated with distinct tissue microenvironments and inflammatory states. So in the next few slides, I'm going to talk about a new study from our laboratory that was published earlier this month in the JCI. And we wanted to see if we could drill down a bit further on mast cell heterogeneity and granularity by creating a new data set where we flow sorted mast cells from 4 aspirin-sensitive donors. We used the 10x Genomics V3 Chromium platform. And we ended up with about 20,000 mast cells across our 4 donors versus a little over 1,000 in our prior data set, which really did give us a much more granular look at mast cell heterogeneity within the polyp. Now, as before, we saw MCTs and MCTCs, but we also found some subclusters within the MCT and MCTCs. When we looked at the genes underlying it, it looked like those subclusters were really driven by activation status, right? So there was this kind of module of genes associated with activation, but also the activation genes were different in the MCTs and the TCs. So in the MCTCs, we saw enrichment for IL-13 and CSF2, GM-CSF. In the MCTs, that activated cluster had enrichment for a lot of enzymes associated with icosanoid biosynthesis, as well as IL-5. When we ran some pathway analysis, we saw sort of a similar picture where our MCT2 and MCTC2 populations were enriched for a lot of transcripts associated with activation. Right, so I think this is a sort of an important concept with single cell sequencing. Sometimes 2 clusters are different cell types. Sometimes they're different states, right? Sometimes that might be activation. Sometimes that might be proliferation. So it's always really important to think about what is driving your heterogeneity. So here it looked like activation was at least one of the major drivers. We also were able to kind of looking for clues as to what might be driving this different populations. We were excited to see that there was one of our hits in the MCTs was this SMAD protein phosphorylation. And we were excited to see this based on some prior mouse work that we've done, linking TGF-beta to mouse mucosal mast cell differentiation. Us and others have kind of seen this link in the past. So we wanted to see if we could dig down on it a bit more and see if this might also be an important signal for human MCT differentiation. Now, one of the challenges that we've faced with doing pathway analysis on mast cells is that a lot of the differentially regulated genes in mast cells across various stimulus conditions, they don't exist in other cell types, right? Things like the proteases they express or receptors like MRGPRX2 that you see maybe in the nervous system and mast cells. So this really, I think, limits the utility of these canonical gene expression pathways. So to get around this, we've really started making our own. So in particular, we found that the MCTs within the nasal polyp were really robustly enriched for a gene signature that we constructed for mast cells that are differentiated in TGF-beta for 7 weeks here. And so this, again, further suggested that TGF-beta might be an important signal here. So when we specifically focus on the proteases that are differentially regulated in vitro by TGF-beta, we can see that there is a really robust downregulation of MCTC-associated proteases. Kinase goes down, cathepsin G goes down, also things associated with generating heparin. And that seems to be kind of happen in this time-dependent manner. The longer the mast cell is sitting in TGF-beta, a day, a week, or the entirety of its development, the more downregulation we get. We also then, when we did intracellular staining on these mast cells that we differentiated for 7 weeks in TGF-beta, so here we're using peripheral blood-derived CD34 cells, we're culturing them in TGF-beta, or with or without TGF-beta, and then just otherwise standard mast cell differentiation conditions, IL-6, stem cell factor, a little IL-33 at the start. Culturing these cells in TGF-beta also gives them an MCT-like protease phenotype at the intracellular level. So it really significantly downregulates kinase and cathepsin G, which was exciting because now it looked like we had a tool that we could use to really start to put the human mast cell subsets against each other and understand how do they differentially influence inflammation. So as I alluded to at the beginning, we saw substantial differential expression of pro-inflammatory mediators in our newer single-cell sequencing data set, full extent here, shown over here. And so we wanted to understand if TGF-beta was sort of a driver of these differences in mediator gene expression. So to test this, we took our in vitro differentiated MCTs and TCs, and we activated them with either IgE cross-linking or IL-33, and then we used multiplex ELISA to measure their mediator productions. So a couple things immediately jumped out at us. One is that there were clear differences between our Ts and TCs and what they like to make, but also there were differences in how the mediator profiles they made in response to IgE cross-linking versus IL-33. So there's clearly the selectivity, right? So our MCTs made more IL-10, for example, after IgE cross-linking, but our TCs after IL-33. Now, this is sometimes a heat map like this can be difficult to really appreciate differences. So I'm just kind of spinning out separately. This is just bar graph to show the IL-5 and IL-13. So you can really see how stark the differences are. So our in vitro differentiated MCTs make a lot of IL-5, very little IL-13. And we see the opposite with the MCTCs. And this really mirrors the gene expression profiles that we see in vivo, where MCTCs may have IL-13 enrichment, MCTs have IL-5. So mast cells are also a really big source of pro-inflammatory eicosanoids, including leukotriene C4 and prostaglandin D2. And these are these really potent lipid-based mediators that are generated from nuclear membrane lipids using various enzymes within the cell. And a lot of these enzymes were very differentially regulated across our subsets. We could also see that when we treated mast cells with TGF-beta, we saw many of the same patterns, where MCTs in vivo are enriched for cystoleukotriene biosynthetic enzymes, as well as COX-1. MCTCs had a higher expression of PGD synthase. In vitro, we could see the same, where TGF-beta really upregulated 5-lipoxygenase, LPC4 synthase, all of these eicosanoid enzymes, as well as COX-1, while downregulating PGD synthase. So ultimately, what was the functional consequences of this? Well, our MCTs that we differentiate in vivo are cystoleukotriene factories. They make massive amounts of cystoleukotriene to the extent that this was actually statistically significant in our MCTCs, but you just cannot appreciate it. It's really swamped out. They also, kind of to our surprise, based on that PGD synthase downregulation, make a lot more PGD2, but this ended up being due to that COX-1 upregulation, which seems to be really the rate-limiting step. But this is in vitro. Really, what we care about with human is what happens in vivo. So we flow sorted our MCTs and our MCTCs based on surface markers that we previously defined in our prior paper. And we could see really the same patterns. The MCTs that we isolate from our nasal polyp make massive amounts of cis-leukotrienes when we activate them compared to the MCTCs. And we saw a trend towards more PGD2, but that did not end up being statistically significant. So we think that probably, cis-leukotriene generation this is probably one of the major things that MCTs are doing within the context of inflammation. So in the last couple slides, I want to just briefly touch on an unpublished study that we're working on right now, where we're using single-cell RNA sequencing to kind of try to understand the transition between a circulating mast cell progenitor and the mature mast cell in the tissue. So for this study, we've used cell hashing to and site-seq for identifying cell surface markers on paired samples from 8 donors, where we took the nasal polyp out of their nose, and then at the same time, we took peripheral blood. From the nasal polyp, we looked at lineage negative CD45 positive cells, and from the blood, we look at CD34 positive cells. When we combine those data sets and do a little bit of data curation, we can see kind of this nice UMAP where we have a lot of our progenitor populations on the left with mast cell progenitors here in pink. This is essentially the same population that Joakim described through single cell sequencing fairly recently. And then all of our mast cells in the nasal polyp on the right. So when we break that up by tissue, we can see that we do actually see a few of those progenitors in the polyp, although not many. There's really ended up being about 3 per donor. We saw something different when we looked at the surface proteins. So a lot of those proteins that are enriched in MCP in the proliferation or in the circulation tend to be associated with the proliferating mast cells in the nasal polyp. So currently we're working on kind of drawing more links between these, but just kind of showing you one of them, CD71. When we look in the nasal polyp and we subset out the proliferating Ki67 positive versus Ki67 negative, we can see that CD71, which is the surface marker associated with circulating progenitors, is very robustly enriched in the proliferating mast cells relative to the quiescent one. So we're really excited on kind of digging into this more going forward. So I am out of time, and I apologize for going 30 seconds over. But for briefly wrapping this up, we're really excited about using single-cell sequencing as a tool to really dissect mast cell heterogeneity. And we find there's these fluid gradients underlie these kind of binary histochemically defined subsets, right? So I think there's a lot more complexity than you can see when you just stain for 2 markers. And we're really kind of excited to use this technology more going forward and continue to learn things. So with that, I'd like to thank the people in my lab who have done all the work, our collaborators, and our funding. So thank you so much.
Thank you very much, Daniel. It was really an exciting talk. And I really would like to start with the questions. There are some more general and some more specific. The first question I would like to ask is whether the proliferating mast cell population in the nasal polyps tissue was a mature cell population or a progenitor cell population?
Yeah, so I see that's timestamped before on the last few slides. So yeah, we think it's really associated with the proliferating mast cells. So we've looked at CD71 as one marker. We've looked at several cell surface markers and intracellular markers, and for a lot of these, we think there's really a link between that circulating mast cell progenitor and the proliferating population we see in the nasal polyp. That this is probably sort of one of the questions in the field, right? The mast cell progenitor that circulates is so incredibly rare. It's a 1,000th of a percentage point of our PBMC pool. So how can this give rise to this massive population in the tissue? And we think that proliferation is really an important amplification loop there.
Okay. And we noticed the IL-17 receptor B expression by mast cells in one of your slides. And this is intriguing. Frequently, however, this mRNA is expressed without being translated. And this is typical in ILC2 cells. So do the mast cells respond to IL-25? So what is your experience?
Yeah, so we have not yet tested that. At the moment, we just sort of think of it as a transcriptional marker of the cell. I think it's an important question. Now, one thing I think is that, that IL-17RB is only half of the receptor for IL-25. They don't seem to express the other half. So I'm not sure to what degree this is going to be something that they're really, functionally going to be responding to. We're sort of trying to work out some systems to test this at the moment. It's difficult to get induction of this gene in vitro. We still haven't really figured out exactly how to do it. It at least somewhat responds to IL-4, but not to a huge extent. So I think that very much on our to-do list, but we don't have that answer yet.
Okay. This is one of my favorite questions. Do mast cells or subtypes thereof ever express chemokines receptor like CCR3 or eotaxin? Do you see differential chemokine or chemokine receptor expression?
So we don't really see much in the way of chemokine receptor expression on mast cells, at least in vivo. If we're talking about humans, amongst conventional chemokine receptors, it's really just CXCR4. That's really the one that we've seen best in the tissue as well as in the circulation. But the other one that we're really interested in working on, and there's a postdoc in my lab who's sort of wrapping up this project now, is actually GPR183, right? So this is a chemotactic receptor for oxysterols. And we think that this is actually quite important. It's on the progenitor, it's on the mature MCT and the nasal polyp. And we think that this actually might be one of the really important signals that they're responding to.
And do mast cells in mastocytosis show these population gradients? And how can that relate to disease severity?
So, I mean, we see gradients of expression. So we have used single cell to look at mast cells in the bone marrow and mastocytosis. And we haven't compared, for example, the bone marrow to the skin. Really interested in doing that, but we haven't yet done that. But within the bone marrow, we can see, instead of kind of having this two-directional gradient where they're going towards kind of kinase versus the MCT phenotype, it really seems to be sort of a single gradient. So we can see, a gradient of maturation, upregulation of kinase, upregulation of cathepsin G, but it seems to be sort of associated with more the maturation of a progenitor to a mature MCTC rather than kind of selectively going in multiple directions. That said, we've just looked at a single tissue, which is the bone marrow, that very well may be different in different microenvironments.
What about tissue aging? How does it change the signature?
Fantastic question. I think there's good data to suggest that sort of the neonatal mast cell is very different from the one that's present in mature tissue. That's, I think, best understood in mice, but even, experimentally, if you grow cord blood mast cells versus growing them from adult peripheral blood, they're very, very different beasts. So, I think that this is also, it's a complex question, right? More complex than it might be on its face, because you have sort of the two things. One is the tissue environment itself is aging. On the other hand, the progenitor source may be changing. So I think, Chris, I don't have a great answer, but I think it's really interesting to think about how these two things are interacting with each other.
And you're using purification of cells and cell sorting, and so there is a question about what are actually the advances in situ single cell analysis regarding mast cells. Do you have any update on that?
Yeah, so we haven't really done a whole lot of work with spatial yet. I think it's very much on the to-do list. I have found that, at least as far as studying mast cells go, often the probe sets that various companies use are not super satisfying. So I was going through, for example, the 10x Genomics, like their new 5,000 gene panel that they've released for Xenium, which I was super excited. And then I saw the one, they have cathepsin, or they have kinase in there, which they say is a marker of intraepithelial mast cells, which, it's not in humans. And there's no tryptase in there. We're not seeing any of the tryptase transcripts. So I think that, the current panels are not actually great for looking at mast cells. We're sort of thinking about how we can put together a custom panel to supplement that to maybe see something. They're clearly there. I would say probably the concentration we would see on a slide is higher than we'd see by flow, because on flow we have to digest them out and we're losing a lot of cells. So I think, a very important question, very exciting question, but I don't think there's a great, answer quite yet.
And this is a sort of a provocative question. So are there any mast cell population that do not express FCF0 receptor?
So I think there's the transcript level and there's the protein level, and that these are distinct from each other. So at the transcript level, I have not seen a mast cell that does not have Fc epsilon R1, with the caveat that we're looking primarily at adult mast cells, right? So I think that it's different when we're thinking about the neonatal window where you might have very, very low levels of expression. Now, from single cell sequencing and also just doing a lot of flow, you know, we've known for a long time that Fc epsilon R1 is stabilized on the surface of a cell by IgE. So I myself have very, very low levels of circulating IgE. I need to use PE to find Fc epsilon R1 on my circulating progenitors. If I try to use anything else, I see nothing. We have other, you know, people in the division that are more atopic, have higher levels of IgE, and on them you can use pretty much any fluorophore to see Fc epsilon R1. So, you know, I think that it is always important to sort of think there's doing one mast cell skin, Fc epsilon R1 is quite low in someone who is, you know, non-atopic. You might see very little expression, but that doesn't mean that transcript's not there.
Yeah. Thank you, Daniel. There are plenty more questions, but we ran out of time. So I think you could answer afterwards then in the chat all the questions that are remaining. And I would pass the presentation to Roma for the next speaker. Thank you very much, Daniel. It was exciting. Thank you again.
Great. Thank you so much.
Thanks a lot, Dan. Our next speaker is Dr. Joakim Dahlin, and he'll be talking to us about single-cell dissection of human basophils. Dr. Dahlin is a principal investigator and team leader at Karolinska Institute in Sweden. He received his PhD degree from Uppsala University and conducted postdoctoral research at Karolinska Institute and the University of Cambridge in the UK. In 2019, he became an assistant professor at the Karolinska Institute and established his own group. He's now docent and principal researcher in experimental hematology. His team develops and applies single-cell omics methods to chart human basophil and mast cell differentiation, to pinpoint how these cells can contribute to maintaining health and how dysregulation or deregulation of these cells causes hematological neoplasms. I hand it over to you, Dr. Dahlin.
Yeah, thank you very much. Let me share my screen. It's a pleasure to give this talk here today. I will talk about basophil and how to dissect them with single-cell omics. All of what I would talk about today is unpublished work. So I'm happy to share this. So this is about single-cell omics-based characterization of human basophils reveals two transcriptionally distinct populations. And the first thing I would like to introduce is single-cell omics. Now, Dan already talked about this, but many people use it and it's become a very popular tool to understand health and the development of disease. And here I just show a resource published in Nature Medicine, where they characterized peripheral blood cells using single-cell transcriptomics. And when I look at this landscape, you know, I see a lot of cells that you can study cellular changes and molecular changes during an infection or any type of disease. But you see lymphocytes of different types, T cells, monocytes, and you see different types of hematopoietic progenitors. But what I'm always missing when I look at thousands of these is the basophil population. And if you go into databases as well, you find a lot of cell types, but always missing the basophils. So this is a single cell atlas. This is another example cell typist. Still no basophils. So to present the basophil, I would say that this is the forgotten cell of the immune system. At least that's what the previous slides would show. They are very rare, so they are about 0.5% of all the blood leukocytes. And for the context of my presentation here today, I also want to introduce that they express CCR3, which is commonly used in the clinic for the basophil activation test. They also express Fc epsilon 1, the IgE receptor. And also for the context of this presentations, it's good to know that they secrete histamine and they have charpent-laden crystals encoded by the gene CLC. One of the reasons why they probably underrepresented or actually missing from most single-cell data sets is that it's very difficult to extract the RNA from human basophils. And I would say it's not really pinpointed why. So the aim of my talk or our study is to put the human basophils on the single-cell omics map. And I can also mention that this talk will be about the human basophil. So if you're interested in mouse, there are a lot of publications, including our own, that characterize the human basophils. And if you're into single-cell sequencing, I would say it's quite straightforward, even though the technology itself can be a bit challenging. So if you're more interested in that, I recommend reading other papers already published. So the goal of our research here is to capture mature human basophils using single-cell RNA sequencing. Another goal that we can achieve at the same time is to investigate basophil heterogeneity. And if we generate this, we also want to make it available to everyone and to make like a user-friendly basophil resource where one can go in and check your favorite gene and how it's expressed among basophils. So let's start with trying to capture the mature human basophils using single-cell sequencing. And before I jump into this, I would like to mention the technique we're using, and we are using concepts from CiteSeq. And this method was published in Nature Medicine and Nature Methods in 2017. And it allows the capture of both the cell's transcriptome and the immunophenotype at the same time. And roughly this is how it works. So you have a cell, you have the RNA in the cell, and you can analyze the transcriptome based on that. What you also can do is you can label antibodies, conjugate them with the oligos, and the oligos will then act almost like a fake gene that you can pick up during the bioinformatics analysis. So then you can quantify how much of this surface protein that is expressed. So what did we do? So we started with the peripheral blood. We started with the buffy coats, so quite a lot of leukocytes, and we isolated the low density fraction of the blood leukocytes using FICO. We then performed our cell barcoding technology and also sorted the cells. So we knew exactly where the basophils are, because since we don't know exactly what they will look like in the single cell resource, we want a tool to trace them very accurately. So we first sorted the unlabeled leukocytes, which will include different types of lymphocytes and monocytes and other cells. And then we specifically barcoded and sorted our basophil population. And these basophils, when we sorted them, we not only added these antibodies with the oligonucleotide, we also labeled them with a fluorescent marker, which allowed us to keep track of exactly the number or the fraction of basophils that we put into the single cell sequencing pipeline and how many we got out of it. We then used CiteSeq, as I mentioned before, but we customized it a bit. Today I will not go into all the details about the customization, but this is really to make sure we can capture all the RNA that they have, because it is difficult. So how do we define the basophil population? So the sorting strategy we used is, first of all, we gate on the side scatter low cells, and eosinophils, for example, are already removed because they are high density cells. So we isolate the side scatter low cells, lineage positive, so we exclude B cells, T cells, monocytes, and then we sort based on CCR3 and FCF1. So a very clean basophil population. And then I said we can measure how many basophils we put in the sample and how many comes out. So we can actually use flow cytometry to track the exact number of basophils. So we have 26% basophils in this sample. So it's uneven, but it's on purpose here, because otherwise you would have up to 1%. And this is what you get out of it when you run the bioinformatics pipeline. So in the end, we achieved 17% of basophils. So there is a specific loss of the basophil population in the single-cell RNA sequencing pipeline. We don't know exactly when they are lost, but we can see a small loss. And the great thing here now, when we have them barcoded, is that we can track them when we visualize them on this UMAP. So we just plot the cells that have the barcode, and we know exactly that these are our cells of interest. And based on other cell type or cell lineage specific genes, we can annotate the rest of the landscape as well. So we have different types of lymphocytes, and we also see some neutrophils here. We can, of course, plot the expression of certain basophil-related markers like CCR3, which we use for sorting. So they are obviously positive, all of them. And also histidine decarboxylase, which makes the histamine. So now we have a method that we can detect basophils using single-cell RNA sequencing. The next question is, what about the basophil heterogeneity? Is it just one population of one cluster or one phenotype of the basophils, or is there more into this? And to address this, we of course need a lot more than just a few hundred or 1,000 basophils. So that was our next thing to dig into. And to do this, we used this experimental approach. We started off again with buffy coats. We extracted the low-density blood leukocytes. We sorted out the basophils specifically using exactly the same strategy as we did before. And then again, our customized site-seq protocol. In this case, we got 6,000 basophils when we produced the first data set. And then the results looked like this. We were quite surprised by this. We got two populations, basophils 1 and basophils 2, we call them here. If you look at the expression of different genes, they are very similar. So most genes are comparable in the gene expression level, but there are a few exceptions like FTL and CLC. CCR3 and HDC, which I showed you before on the entire basophil population, is very similar. So this was, to us, a very surprising observation that would be two transcriptionally distinct basophil populations. So we asked ourselves, is this something we can reproduce in more than this donor? And we also consider, okay, is this something with the isolation method that could artificially produce this? So we thought, okay, we need to address this. So we wanted to generate additional data sets. So in this case, we made 3 new data sets from 2 independent donors. We changed the method to extract the cells from FACS to MAX, and we combined the single-cell RNA sequencing with the large-scale immunophenotyping, and in this case, using 140 different antibodies. This is the result. So you see the same 2 populations again, basophils 2 and basophils 1. It's evenly dispersed in the different donors. So there are no donor effects that give rise to the 2 populations. They actually exist in both. And you can also process each sample or each individual sample by its own, and then you still see the 2 populations of basophils. So this is very reproducible. The differentially expressed genes remains the same. So FTL, CLC is there. And then we thought, okay, what can we do with this immunophenotyping? So since we do this large-scale characterization, we can of course look what gene or what surface markers are expressed on the entire basophil population, but we can also look what is different between basophil 1 and basophil 2. And the first thing we noticed is that we see no signs of activation. So CD63 is the same. Another marker of activation in basophils is CD69, also remains the same. There's a study from a few years ago, 2020, that used mass cytometry to identify different clusters of basophils. And they distinguished some based on the expression of CD16, CD244, and Fc-epsilon-1. But to us, they all remain the same, and we couldn't use these markers in any way to separate and distinguish different types of populations. But we still see the transcriptionally very distinct basophil populations. So among actually all the markers that we characterized, the 140, there is not a single marker that's significantly different between those. So we're quite sure that both are basophils. The one that stood out is Fc-epsilon-1. It's not significant, but it's the one that is the most different, and it's quite similar. And then we also ask ourselves, so does a deeper characterization of the RNA allow further insights into basophil heterogeneity? So this was a bit of a crazy experiment that we did, got an opportunity to do. So we did full-length RNA sequencing using nanopore sequencing. So this is a method that was published a few years ago, and it was great for us because we could repurpose the remaining cDNA that we have in the freezers. So what we did was we did this nanopore sequencing method, and then we still distinguished the two populations of basophils. So this basophil 2 here and basophil 1. We see differences in FTL and CLC, exactly like before. But we also saw this small population of, or small cluster of basophils that we here just named basophil 1 ONT. So this was specific for this data and for this data set. And there are two long non-coding RNAs that highlight this basophil 1 ONT cluster. So we don't know what it means, but it's clearly different in both donors. We also recognized that we could detect additional genes that we didn't see with a short-read Illumina sequencing protocol. So this is just one example. T-span1 is quite different and higher in basophil 2, and you cannot recognize this at all in the Illumina data. So it gives additional insights to help us understand basophil heterogeneity. So the last thing we wanted to do was to create a user-friendly basophil resource to allow everyone to dig into this and start to understand what these two transcriptionally distinct populations are doing. It will be available soon, released soon. It looks something like this as of now. So you can go in and plot your favorite gene or your favorite antibody or surface marker. We will also make sure to apply a CC-BY license. So if you want to reuse the data, you can put it in your own publication later on. So the conclusions from now, we capture the human basophils using single-cell RNA sequencing. We see the two transcriptional distinct populations, and we show that they have a very similar immunophenotype. And then we have produced this resource to make it available through a web interface. And of course, this is now basophils in the healthy setting. We would, of course, want to know in the future, okay, what happens in the disease settings? So looking at your favorite disease and see how the dynamics change would be very interesting. There are, of course, a lot of people involved in the work. I would like to especially say thanks to Sofia. She's the one who's been driving this project in the group. All the hard work, both on the experimental and bioinformatics side. I also like to thank all the other members of the team. And there are also other groups that have been involved, of Gunnar Nilsson, and a lot of help from SciLife lab here in Stockholm to help with nanopore sequencing. A lot of discussions with Anand and Lars at A-Star and the University of Copenhagen on these, understanding these populations, and all the funders. And thank you for your attention.
Thank you, Joakim. Thank you very much for this talk on basophil and also on the difficulties of dealing with this population. Indeed, we are interested in understanding whether this diversity, this heterogeneity in basophils is also present at the in the tissue and under inflammatory or disease situation. What is your feeling, your hypothesis in diseases like urticaria? Do you think, for example, that the basophil phenotype or the transcriptome of basophil could reveal heterogeneity?
Yeah, I'm sure it will. Yeah, I think so. The problem is still to actually identify them, to pick them out, because I think in the most data sets that we even reanalyzed, knowing exactly what the basophil looks like and doing everything to pull them out, it's still actually difficult to pull them out from other people's data sets. Sometimes we pick up rare examples of them, but I think one needs to also customize the protocols to be aware of that you want to study basophil specifically, because if you don't do that, you will lose them, and you will analyze all other cells first.
And do you know if there are resident basophils in normal tissue, like you have for eosinophils?
No. I don't know is the honest answer. And I think what it makes it so difficult is that they are, of course, in the blood. So how can one make sure when you extract the tissue and extract cells that you're not extracting the cells that are in blood vessels or something? So I think that's a huge challenge when studying basophils at peripheral sites. But it's, of course, something we'd like to look into, but yeah.
And one question is a technical question. So because basophils express actually some RNAs that you find also in eosinophils, do you think is an issue? Is that something that is interfering?
I suppose it is. There are also other challenges with the basophils, because from what we can see, it looks like not all the RNA is processed. And I think that's the reason why a lot of people have missed this.
Yeah.
So even though they pick it up, they don't see it because they look for processed mRNA.
And you described the two populations in the blood, basophil 1 and basophil 2.
Yeah.
Are they morphologically different, these two populations?
Yeah. And we have struggled for maybe 2 years trying to pull them out. Our goal was to really make sure that we could sort them, but we didn't find a single surface marker that was different, so we couldn't use that. The second thing is we, of course, looked at the genes that were different between them, and everything that we find, more or less, is intracellular. So even if we could sort them out, they will not be alive. And then we might have this discrepancy between RNA and the protein. So how do we know we actually sort out the right cells?
And where do you think these two populations of basophils actually origin? So are they a product of an heterogeneity that is created in the blood, or is already developmental?
Yeah, it's a good question. We don't really know where it comes from. Before we got questions about activation, and I really don't think that's it. It could be that some, are easier to activate. That we don't know, but don't think it's a matter of activation. It could be that it's different developmental stages, that some are almost completely mature and the other one represents the terminally mature basophils. But also we don't really see this it's almost a gap between them, and there are very few transitioning cells in the kind of space between them. So, good question.
Have you tried to culture these two basophil populations?
We have tried to culture basophil progenitors, have been our goal. The difficulty still here is that we cannot culture the two populations separately. So what would happen is that we can put them both in culture and not knowing which one is which, and then analyze them later. And we haven't tried that.
Okay. Thank you very much. Joakim, you have more questions in the chat. I would ask you to answer the question, and then we come to the last speaker, and I will give the word to Roma again for the presentation. Thank you very much.
Thank you. The next speaker is Dr. Netali Ben-Baruch Morgenstern. She's going to be talking to us about single-cell dissection of human eosinophils. So Dr. Ben-Baruch Morgenstern joined the Rothenberg Cured Lab after completing her PhD at Tel Aviv University, where she studied the regulation of eosinophil hematopoiesis, chemotaxis, and activation. Her current research explores cellular composition, molecular characterization, and disease-promoting mechanisms in eosinophilic esophagitis and eosinophilic gastritis. She aims to utilize single-cell RNA sequencing and protein analysis to create publicly available datasets and comprehensive analysis to identify new therapeutic targets for treating eosinophilic gastrointestinal diseases. I hand it over to you, Dr. Ben-Baruch Morgenstern.
Hi, thank you so much. I'm really honored to be part of this panel. Just a second, okay. So, thank you very much for this for the invitation to present this data. I'm really honored to be part of this panel, and I will present today on single-cell dissection of human eosinophils. So eosinophils are granulocytes. They originate from the bone marrow, and upon maturation, they release into the peripheral blood when they infiltrate during homeostasis to various tissues and serve homeostatic roles. They also participate in various inflammation, including allergic inflammation, infection, hypereosinophilic syndromes, and autoimmune disease. Under both homeostatic and pathological condition, the interaction between eosinophils and other cellular population in the tissue exhibit distinct bidirectional effects, varying based on the specific milieu and leading to the heterogeneity in the eosinophil population. In the last 50 years, scientists defined eosinophil heterogeneity in multiple ways. One of the first ways to define eosinophil heterogeneity was based on their density, when nomadens, or before that even they called it hyperdens, described the resting cells, while the hypodens described the activated as they release their granules. Later, surface markers started to be helpful in order to define the heterogeneity, such as CD69 that define if the eosinophil is activated or not. CD11B and CD62 define the adhesion and migration potential, and so on. Another way to define eosinophils is based on their localization, if they're coming from the circulation, or they are tissue resident, or they infiltrate in eosinophils into the inflammation. How they drive, based on which cytokine drive them, if they are driven by IL-5, which can promote their differentiation, or IL-4 or IL-13, which define their activation status in allergic inflammation. Electron microscopy helped to define morphological variants, if they are degranulated or intact. Developmental stages were defined based on their transcription factor status. Disease-specific heterogeneity, and protein subtypes. By staining granule proteins of eosinophils, we can define to which granules they are positive and to divide them based on that. And one of the recent one is the transcriptome subtypes, which its gene expression pattern that can reflect also functional differences. With that method, many scientists had found an issue because due to eosinophils susceptibility and increased levels of RNases, this method was a bit limited, and I will elaborate. Single-cell RNA sequencing in the last few years become extremely popular method for scientists to examine cell population and to examine disease status compared to control. And one of the most common method that was publicly available was the droplet base, also known as 10x. Scientists that were interested in eosinophils very fast noticed, similar to what we saw in Basophils, very fast noticed that eosinophils are not present in the tissue. Even if these biopsies were obtained from patients that is known that have eosinophils in the tissue by other means of flow cytometry or immunohistochemistry, in the dataset, we couldn't find eosinophils. In parallel, or a bit later even, another method of single-cell RNA sequencing become commercially available is the gravity-based. The gravity-based is different from the droplet that is used gravity instead of shear stress, and that is why it's more gentle compared to the droplet. Based on the gravity, it pulls the cells into the microarray and sequence it cell by cell. The limitation of this method is that because of the size of the microarray, big cells, such as the structural cells, such as epithelium and fibroblast, cannot go inside of the well, and that's why we get some kind of artificial enrichment of the leukocytes in the tissue, and that's when we are comparing between the UMAP that we get from the same tissue. In that case, this is the esophagus. We can see completely different UMAPs. However, because in the gravity-based we can actually see eosinophils, many scientists that pursuing after eosinophils data continue to use on the gravity base. One of the most comprehensive single-cell transcription profiling of eosinophils was based on the gravity method, published recently in Nature by Gartner et al. They use mouse model, alpha transgenic mice, and identify from different organs, from different disease status, and from homeostasis, at least 6 eosinophil population. One of the population was the active eosinophils that was specific to the gastrointestinal, and by using the single-cell RNA sequencing, they could conclude on the functional and to create hypotheses and investigate them. And they found that these active gastrointestinal eosinophils enhance bactericidal activity, defend against pathogen, regulate immunity via PD-L1, and play dual role in colitis driven by IL-33 signaling and NF-kappa B activation. So based on the single-cell RNA sequencing, we can get knowledge and we can gain knowledge, and we can conclusion from this beautiful method. So what about human eosinophils? So if you're going inside of PubMed and you open PubMed and you're searching for human eosinophil single-cell RNA sequencing, we cannot find many manuscripts. In fact, when I prepared the presentation, I could find less than 5 publications. And the reason why is because it's likely because many scientists spend so much sources in order to prepare the data set, but they based on the droplet and not based on the gravity. So what we were interested to do is to utilize the gravity base in order to first characterize peripheral blood eosinophils in type 2 inflammation. So our model was the EOE, and we were interested to investigate what is the difference between peripheral blood eosinophils that obtained from active EOE patient as a model for type 2 inflammation compared to control individual. So again, we used the gravity, and indeed we succeeded to identify the eosinophils. And when we subset these eosinophils and looked on the cells the eosinophils that obtained from the control and the EOE, we could see very homogeneous population. And you can see that here, every dot is eosinophils, the red coming from the EOE, the blue coming from the control. And we can see that they are all mixed together. And when we perform differential expressed genes, we could see that there are no differential expressed genes in active EOE compared to control. In parallel to us, two other manuscripts came, and they investigated also the peripheral blood eosinophils in other two type 2 inflammation diseases. One of them was from patients with asthma versus control, and another one from chronic rhinosinusitis. And both of us, similar to us, demonstrate that the eosinophils that obtained from the type 2 inflammation were extremely similar to the eosinophils that isolated from the control. So we concluded that peripheral blood eosinophils obtained from patients with type 2 inflammation and control individuals exhibit similar properties. Then we hypothesized that peripheral blood eosinophils are relatively non-activated. If they're so similar to healthy controls, it's likely that they are not activated. In order to address that, we again focused on the eosinophils from the peripheral blood and perform functional and biological process analysis. And we identified that eosinophils enriched with biological process that associate mainly with leukocyte migration and metabolic processes. And that's why we concluded that peripheral blood eosinophils are relatively non-activated and enriched with pathways associated with migration and homeostatic metabolic process. Next, we hypothesized that peripheral blood eosinophils undergo transcriptional changes enriched for activation pathways upon infiltration into the tissue. In order to address that, we used our dataset from the peripheral blood that we created, and we integrated it with publicly available dataset that was published in Science Immunology from the Christopher Love Group. And this is esophageal biopsies from active EOE patients. The reason why we could do that is because both datasets were gravity-based, and has been done by the same method. We integrated them computationally and created a big dataset of esophageal tissue and peripheral blood that we were able to observe the eosinophils. Subset in the eosinophils allow us to see that in the peripheral blood, we can see one united population similar to what we saw previously. In the esophageal tissue, on the other hand, we saw two distinct populations. One of them was peripheral blood-like. It was similar to the population of the peripheral blood. Transcription is similar. And another one was esophageal exclusive that was not present in peripheral blood. So our conclusion was that two distinct eosinophil populations exist in the esophagus. One is transcription similar to the peripheral blood, whereas the other is exclusive to the esophagus. Then we were interested to understand what the function of these two distinct populations. When we're looking at the marker genes of the peripheral blood genes, we can see that they enriched with 62 marker genes. The esophageal exclusive were enriched with 659 genes, and of course, they were not overlapping between these marker genes. When we perform analysis on the peripheral blood eosinophils from the 62 marker genes, we can see that peripheral blood eosinophils are enriched with biological processes that associate with respiration, ATP synthesis, and cellular respiration. The functional analysis demonstrate that they are focused on respiratory metabolism. On the other hand, when we looked on esophageal exclusive cells, we can see that they are enriched with signaling pathways, including IL-1, TLR, TGF-beta, NF-kappa-B signaling, and MHC class 1 antigen presentation. And the biological processes demonstrate that these are activated leukocytes. Again, when we are comparing our results with a functional analysis of eosinophils that obtained from nasal polyps in chronic rhinosinusitis, they compare the eosinophils in nasal polyps compared to peripheral blood as well. They saw something that is extremely similar to what we saw. The eosinophils that obtained from the inflammation site they were increased with anti-apoptotic genes. They were increased with NF-kappa B signaling, similar to what we saw. And they are increased with cytokine mediator signaling pathways, again, similar to what we see. And overall, the biological processes was inflammatory response, cytokine mediated signaling pathways, and negative regulation of apoptotic process. And overall, the functional was cytokine signaling in the immune system and signaling by interleukin. So collectively, these two analyses suggest that eosinophils undergo activation in the tissue. Then we hypothesized that esophageal eosinophili are mostly transcriptionally different from other inflammatory cells that accumulate in the tissue during active EOE, the mast cells. And in order to investigate that, we looked on mast cells and eosinophils in the total esophageal tissue. Eosinophils express 898 marker genes, and mast cells express similar levels of 1,098 genes in the esophageal tissue. Only 161 of them, of the marker genes, were shared between the two populations. So we can say that indeed, esophageal eosinophils and mast cells seems to be transcriptionally different. Then we wanted to investigate the function and to compare between the function of mast cells to eosinophils in order to examine if they have different function in the tissue during active EOE. So again, we started from the marker genes of each population, but this time we only took the significantly enriched pathways, functional pathways in each population based on the marker genes. Eosinophils were enriched with 56 functional pathways, and mast cells were enriched with 36. 11 of them were shared. The sharing signaling included signaling by interleukin, specifically by IL-4 and IL-13, and antigen presentation. Eosinophils were uniquely enriched with functional processes, including DAP12 interaction, Fc gamma R3A dependent phagocytosis, IL-10 signaling, TLR cascade, and detoxification of ROS, while mast cells were uniquely enriched with metabolism of lipids, synthesis of prostaglandin, unfolded protein response, and Fc epsilon mediated MAP kinase activation, as we expected. So eosinophils appear to be uniquely involved in DAP12 interaction, Fc gamma trigger events, immunoregulation, and IL-10 signaling in the esophagus. The next step for us was we aimed to define the genes highly specific to esophageal eosinophils. So again, we started with eosinophils marker genes with the 898, but this time we applied extremely stringent criteria in order to identify the genes that are extremely enriched in eosinophils compared to the other esophageal cell population. That means that the other cell population hardly any express, if any at all. And here you can see that and get the impression in the heat map that eosinophils are extremely enriched with these genes of signature. We identify only 12 genes that actually pass all these filters. And among these genes, we can see genes that we expect to be there. We expect to see CLC, we expect to see CCR3, we expect to see CABP. All of them are marker genes that known to be marker genes for eosinophils even before this signature. However, in addition to these 3, we have other 9 genes that not necessarily in the past were linked to eosinophils. For example, the transcription factor OLIG1 and 2, the inhibitory receptor VSTM1, the histamine receptor 4, and the succinate receptor, and the other genes, that all of them are under study in the context of eosinophils and for sure deserve much more appreciation in the future. So one of the genes that caught our eyes was the succinate receptor. And the reason why it caught our eyes is because when we are focusing on the esophagus, we can see that succinate receptor is significantly enriched in eosinophils compared to the other esophageal cell population. when we are looking specifically on the eosinophil population, we can see that not only it's enriched in the esophagus, but it's also strongly expressed in the esophageal exclusive eosinophil population compared to the peripheral blood eosinophil population. So the succinate receptor is a 7-transmember protein, G-coupled protein that express specifically by the eosinophil in the esophagus. Upon the interaction with succinate, it can stimulate various pathways. The source for succinate is coming from bacteria that can express succinate. The normal respiration cycle of each and every cell of the Krebs cycle also have the product of succinate, and the exposure for food additives that are specifically the umami taste. Upon interaction between succinate and its receptor, one of the signaling pathways that it stimulates is the ERK phosphorylation, and it has been shown to induce type 2 immune response through the activation of TAF cells in human and also mast cell maturation in murine. We also saw, as part of the research in our lab, that when we take peripheral blood eosinophils that in the transcript level do not express succinate receptor, and we co-culture them with esophageal epithelial cells for 7 days, we see that the eosinophils that were co-cultured increase the expression of succinate at the transcription level. In addition, when we are taking EOL1, that we examine EOL1 cell culture, and we take the naive, we see that at its transcript levels, they do not express succinate receptor. However, when we co-culture the EOL1 for 7 days with esophageal epithelial cells, we see a fundamental increase in the succinate transcript expression. So, due to the esophageal inflammation, there is likely increase in level of succinate in the tissue. We have much more cells, many inflammatory cells enter into the esophageal tissue and they are activated. So it's likely that we have increased level of succinate even just from the normal Krebs cycle. In addition, we have increased expression of succinate receptor by active eosinophils in EOE and by peripheral blood eosinophils in EOE and naive eosinophils following in the interaction with esophageal epithelial cells. So our hypothesis was that succinate will promote phosphorylation of ERK in EOL1 cells that were co-cultured with esophageal epithelial cells for 7 days. So if indeed EOL1 express succinate receptor not only in the transcript level, but also in the protein levels, the introduction of succinate to the EOL1 following co-culture will promote the phosphorylation of ERK. So that's what we wanted to examine. And in order to do that, we performed flow cytometry analysis or phosphoflow. And you can see here that in the first row, we can see the naive EOL1, and in the second row, we can see the co-culture EOL1. And as I mentioned before, the EOL1 do not express succinate receptor in the transcript level, while the co-culture EOL1 do express succinate receptor in the transcript level. In the left is the non-stimulation, in the middle is the positive control with PDBU, and the right is the experiment. We introduced succinate to both of this culture, and we saw that only co-culture EOL1 indeed demonstrate increased phosphorylation following succinate stimulation. So if I'm trying to combine all the things that I showed today and to give some kind of key take-home messages, we can start saying that human eosinophils can be subject to single-cell RNA sequencing using gentle methods such as gravity-based method. The commercial versions are Rhapsody and Honeycomb. Gravity-based single-cell RNA sequencing of esophageal biopsies obtained from EOE patient identified between 100 to 1,000 marker genes per population. Specifically in eosinophils, we saw approximately 900 marker genes for eosinophils. In contrast, when we are looking on droplet-based sequencing, even a recent EOE atlas that removed all the thresholds in order to find these eosinophils, they could identify only approximately 30 markers genes for eosinophils. So for sure, we can say that the gravity base is likely to be the most preferred method to examine eosinophils. 1.5% of eosinophils marker genes, 12 out of the 898, are highly enriched in eosinophils compared with other esophageal cell population. These genes highlight the potential importance of succinate receptor and succinate signaling, histamine receptor 4 and the histamine signaling, and VSDM1, which is inhibitory receptor. A subset of esophageal eosinophil marker genes overlap with esophageal mast cell markers, but the majority of them were different. Functional analysis of esophageal eosinophils revealed that they are involved in DAP12 interaction, Fc gamma triggered events, immunoregulation, and ALT signaling compared with mast cells. Conversely, mast cells are involved in unfolded protein response, Fc epsilon, R1 mediated MAP kinase activation, and metabolism of lipids. Eosinophils isolated from type 2 inflammatory sites, including nasal polyp and esophagus, are functionally enriched with cellular activation and cytokine signaling associated gene. In contrast, blood eosinophils are homogeneous and relatively non-activate and exhibit enrichment with genes associated with migration, endocytosis, and metabolic process, and show homogeneity when comparing cells from type 2 inflammation disease subject and control individuals. And of course, I want to thank to my mentor, Professor Mark Rothenberg, and I want to thank to my colleagues that are co-authors on the manuscript, Mark Hochman and Michael Kotlier, and to the rest of the Division of Allergy and Immunology, and of course to the Cured Foundation that allowing to this research to happen. Thank you.
Thank you, Netali. Thank you very much for your talk. We have some questions, and I would like, and we have only 4 minutes for questions, but all the questions that we cannot answer online, we can send the question as well as the contact information of the person asking, so that you can actually answer the question then per email. So gravity-based single cell sequencing, did you use it also for mast cells? And do you think that there is a difference in using these techniques when you look at eosinophils that are activated versus resting ones? So it's a different output or outcome from using these techniques in different conditions.
So the manuscript that we published in collaboration with Dr. Dwyer was based on the 10xG. When I'm comparing between the gravity base and the droplet base, the number of marker genes and the depth of the method is likely better in the 10x genomic. So you can find better depth and transcriptions in the 10x. However, when you don't have the choice, I believe that the gravity is really good method in order to identify all the susceptible cells. So it's all depend on the question that we are trying to ask. If we are trying to understand what is the connection between multiple leukocyte population and some of them that are too susceptible in order to be identified on the 10x, for sure I would go to the gravity. But if I'm interested to analyze and to go deep on a specific subpopulation without the context of susceptible cells, I will go, of course, I will go to the 10x. And,
okay.
Yeah.
Thank you. And so you show that blood-derived eosinophils are actually resting or not, while the one in the esophagus in inflammatory conditions are enriched in cytokine signaling. So do you think that blood-derived eosinophils play no role in driving type 2 inflammation in AOE?
I think that they are recruiting from the peripheral blood. However, they are becoming activated and can play a role in EoE and in any other type 2 disease. It's only when they enter into the tissue and have all the inflammatory milieu and they are changing the transcription and become more activated. So if we will isolate peripheral blood eosinophils without the right stimulation, they will not become activated. They will stay resting.
Okay, last question. Succinate can promote Th2 responses in some cells like tuft cells in the context of helminth infection. Have you looked at the expression of Th2 cytokines in eosinophils that upregulate the succinate receptor?
So,
Short answer.
Sorry, based on the single cell RNA sequencing, eosinophils do not express Th2, Th1, or Th17 cytokines. We didn't check that in the protein level. This is an ongoing project that we are hoping to address what is the role of succinate in eosinophils.
Okay, thank you, Netali. Thank you very much. I would close the session, but before doing that, I would like to thank the presenters for the really inspiring talks. I also would like to express my gratitude to the attendees and thank them for the questions, as well as I would like to thank Kira and Kati for the excellent organization of the session. Just, I would like to remind you that the full webinar recording will be available on demand, and attendees should keep an eye out for the post-webinar evaluation. And if you want to apply to the IS membership, was displayed before a QR code that you could scan. And I really would like to thank you again and see you by the next session. Thank you very much. And to everybody, thanks.